Okay, everybody, this is Chapter 8, dealing with bacterial genetics and biotech. We're going to talk about what plasmids are and how they reproduce. We're going to talk about some biotech techniques such as PCR DNA sequencing, um, the use of proteomics a bit, and we'll talk about the central dogma, transcription, translation, and we'll talk about gene regulation as well. Here we're going to describe the structure of a bacterial genome and talk about how it differs from a eukaryotic one, and talk about what a plasmid is and how it spreads antibiotic resistance. So the genome of E. coli is composed of DNA, which is not going to be associated with histones in a prokaryote. Okay, there are going to be proteins associated with it, but they're not histone proteins. But the DNA is somewhat condensed, okay, especially when the cell is not undergoing binary fission, okay. There's hundreds of genes on this single chromosome, and it's a circular chromosome, and it's a double helix that follows the base pairing rules, the Chargaff rules, right? The amount of G is equal to the amount of C, and the amount of A is equal to the amount of T, because of the base pairing that occurs across the double strand, right? A always pairs with T, G always pairs with C. Okay, so the two strands are anti-parallel, meaning they run in opposite directions, as shown here, and they're complementary. Okay, bacterial genomes are a little different from eukaryotic genomes. You can see here a section of an E. coli genome, and these include coding genes in green and non-coding sequences in purple, and then a section of the human genome where the human coding genes are interrupted by the yellow regions, which are called introns, and then you've got genes in green, and then you've got these areas of DNA that we're not really clear on what the sequence is. So you can see that the economy of information in the bacterial DNA is much more extreme than in human DNA. And the reason is there are fewer base pairs to work with, and it's a smaller organism. We're also going to find out that in the bacterial genome, there are usually a series of genes that are transcribed in unison off of a single message, and translation happens at the same time as transcription does, and it all happens basically in the cytoplasm of the cell. Okay, so they're not separate processes. The minute the message is made by the RNA polymerase, the ribosomes latch on to that message and begin translating it. We'll also find out that in bacterial DNA, genes can be on either strand, okay, whereas in human or eukaryotic DNA, they tend to be on one strand, okay. So the operon is basically the, the functional unit under which a cluster of genes are expressed, okay. The information that determines when a gene is expressed in the DNA control sequence is the very front of it, which is called the promoter. It's like a gatekeeper to the gene. The genes are arranged in a series, and the unit is called the operon. The single message that's made from the operon has all the genes in it, and they're ready for translation. Okay, so let's take a look at what an operon is about.
An operon is a series of genes in tandem that are controlled by a single promoter. Gene expression begins when RNA polymerase binds to the promoter and transcribes the genes, all into a single mRNA molecule. The genes of an operon typically encode proteins that work together. Here, MDTA, MDTB, MDTC, and MTDD encode proteins of an efflux pump that exports antibiotics such as novobiocin from the cell. Gene BAES encodes an environmental sensor, and BAER encodes a regulator that binds to the promoter and upregulates the expression of the genes in the operon. The DNA sequence specifies messenger RNA and proteins. A variety of sequences in the DNA and mRNA define the landscape of an operon. Here is a closer look at an operon with two genes, A and B. Both strands of the DNA are shown. One DNA strand is the sense or coding strand. It contains a base sequence identical to the mRNA produced from the operon, except that DNA contains the base thymine in place of uracil. The opposite strand is the complementary template strand. RNA polymerase binds to conserved regions of the promoter called the -10 and -35 regions. Binding here directs the polymerase to start transcription just downstream at nucleotide +1. The -35 and -10 sequences are 35 base pairs upstream and 10 base pairs upstream of the transcription start site. RNA polymerase creates an mRNA strand complementary to the template strand. At the end of the operon, a transcription terminator region causes the RNA polymerase to detach from the DNA. Portions of the mRNA are translated into proteins. Each coding sequence of the operon begins with a start codon and ends with a stop codon. At either end of the mRNA are untranslated regions, the untranslated leader and the untranslated trailer. Ribosome binding sites are also untranslated. They direct the ribosomes to the correct locations for translation to begin.
Now, bacteria don't necessarily only have one piece of DNA in them. Some bacteria have extra chromosomal pieces of DNA that are called plasmids, which have the ability to reproduce on their own schedule and frequently carry genes that confer either virulence factors, which are characteristics that help the bacteria to cause disease, or they carry on them genes for the resistance to certain antibiotics, or sometimes both. Okay, they're usually smaller than the chromosome. F plasmids, F stands for fertility, carry genes for conjugation, which is the bacterial counterpart of sex, while R plasmids carry genes for antibiotic resistance, and sometimes you can get plasmids that carry both. Okay. The idea behind the F plasmid is that when the bacteria get near each other, it has a series of genes that codes for a long hollow tube that'll be produced. It'll extend from one bacterial cell to the other, and that's a tunnel through which the conjugating bacteria can send a copy of its genetic material. For instance, a copy of the plasmid DNA can crawl through there, and then when the pilus breaks, both bacteria carry the traits that are on that particular plasmid. As a result, the traits that are on plasmids can reproduce or be spread through a bacterial population very quickly, and even in many cases between different bacterial strains. Okay? You can see here an example of the plasmid and the chromosome in some different bacterial types. Agrobacterium tumefaciens is a bacteria that infects plants, produces something called a gall, okay, big swollen area in the plant, like a plant tumor. You can see that the plasmid, the TI plasmid, is around 2840, and the bacterial chromosome is linear in this case, and is about 2 kb, kilobases. Okay, the entire genome about 5.6 kb. Okay, so that's how you read it. Bordetella burgdorferi, which causes Lyme disease and is carried in ticks and is transferred by a tick bite. Again, the genome is about 911 base pairs, and then there's a couple of plasmids with sizes between 9 and 58 base pairs, so little teeny guys, right? These are the chromosomes, right? This would be linear, this would be circular, and these are the plasmids, okay? E. coli, 4.6 kb circular chromosome, may or may not have a plasmid in it, okay? And Mycobacterium tuberculosis, which is the agent of TB, 4.4 kb may or may not have a plasmid in it, right? If it's blank, it just means that normally we don't find a plasmid in it, but that doesn't mean it can't pick one up, okay? One of the ways that organisms change through time is through a process called mutation, in which changes in the information in the DNA occur as a result of exposure to things like chemical mutagens or ionizing radiation, such as X-rays or UV, okay, or interaction with something like a bacteriophage in the case of a bacterial cell, or a virus in the case of a eukaryotic cell, okay? In addition to the fact that reproduction of DNA is not a perfect process and mistakes occasionally happen that don't get corrected, all right? These changes can either be good or bad or neutral, meaning if they're good, they're adaptive. They make it more likely that that organism is going to live long enough to breed. If they're bad, they make it less likely that that organism is going to live long enough to breed. And if they have no effect, they'll have no impact. Natural selection determines whether or not these changes are beneficial or harmful or neutral. And as a result of that selection pressure, that genetic information becomes more prevalent in that population and is spread to the offspring, okay? And over many, many, many generations, this drives a process known as evolution, which results in new species becoming derived from an ancestor, okay? So we're going to talk about DNA replication, different types of DNA replication. We'll talk about what DNA polymerase does during DNA replication. So let's begin with a discussion of DNA replication.
In most bacteria, replication of a chromosome begins at a single defined DNA sequence called an origin, or ORI-C in E. coli, and ends at a sequence called a terminus. During the first phase of DNA replication, called initiation, enzymes open up a replication bubble at the origin. Enzymes that synthesize DNA can connect nucleotides only in a 5' to 3' direction. That is, every newly made strand begins with a 5' triphosphate and ends with a 3' hydroxyl group. The two parental strands of DNA are antiparallel, which is why the new DNA synthesis will occur in opposite directions. A short RNA molecule, called a primer, is synthesized as a starting point for DNA synthesis. DNA polymerase requires an existing 3' hydroxyl group on which to add nucleotides. But primase, the enzyme that adds the primer, does not. The next phase is elongation. The helix continues to unwind as an enzyme called DNA polymerase III adds DNA nucleotides in the 5' to 3' direction away from the origin. The sites of helix unwinding and new strand synthesis are called replication forks. A circular bacterial chromosome replicates bidirectionally around the chromosome. If polymerases can synthesize DNA only in a 5' to 3' direction, how are both strands of a moving replication fork synthesized simultaneously? At each fork, one strand is synthesized continuously in a 5' to 3' direction toward the fork. The other strand needs to jog back in intervals, each time starting from a new RNA primer. The smoothly elongating strand is called the leading strand, whereas the jogging back strand is called the lagging strand. Note that each replication fork generates a leading strand and a lagging strand, oriented opposite to the other pair. The fragments of DNA that form as the lagging strand is synthesized are called Okazaki fragments. Late in DNA replication, when most of the complementary strand has been elongated, the RNA primers need to be replaced with DNA. The RNA is removed by an enzyme that specifically recognizes RNA-DNA hybrid molecules and cleaves away only the RNA strand. An enzyme called DNA polymerase I fills in DNA nucleotides. The remaining gaps are connected by DNA ligase. Completion of DNA synthesis requires replication at the defined termination or ter site, located on the opposite side of the molecule from the origin. In this termination phase, the DNA helix is completely duplicated and replication stops.
Okay, we're going to talk about mutation next, which is the driving force for natural selection. And we'll explain how mutations occur and different ways that the cell can repair this damage. A DNA molecule is subject to potential mistakes during either its use when the cell is in interphase, where it's simply doing its job, or during the time that it's reproducing. Okay? In fact, the more often that a cell reproduces, the more mutations will take place, because the reproduction of DNA is not a perfect process. Okay? Fortunately, most of the time these changes get fixed before they become passed on to the offspring, but often they get missed, and those changes then end up being passed on to the offspring, and as we stated before, they can either be beneficial or detrimental or neutral. Okay? A mutation can affect the coding region of a gene, the part that contains the protein building instructions, or it can affect the regulation of the gene, okay, which is the part of the gene that controls when and where the transcription will occur. Okay? What causes mutation? Well, like we said before, problems with DNA reproduction, exposure to mutagens, whether they be things like ionizing radiation or exposure to carcinogens, chemical reagents that can oxidize bases in the DNA, or actually attach themselves covalently to the DNA. And then when we attempt to repair the damage by detecting error in the double helix, sometimes the damage is repaired properly and sometimes it isn't. Okay? So let's take a look at base substitution mutations and insertion and deletion mutations.
A mutation occurs by base substitution when an incorrect base is incorporated into DNA. Some base substitutions occur because purines and pyrimidines exist in two structural forms. The most common form results in base pairing between adenine and thymine and between guanine and cytosine. However, the hydrogen atoms can move to form a base with altered hydrogen bonding properties, creating a tautomeric shift. When a tautomeric shift occurs in adenine, the adenine can bond to cytosine. A tautomeric shift in thymine allows it to bond to guanine. This error in DNA replication is passed on to the cell's progeny. The change in a single nucleotide in the DNA results in a change in the corresponding nucleotide in messenger RNA. The change in the codon can result in a different amino acid being incorporated into the protein. The nucleotide sequence in DNA determines the nucleotide sequence in messenger RNA and, consequently, the sequence of amino acids in a protein. A mutation in the DNA can result in a change in the amino acid sequence of a protein. One type of mutation that can occur either spontaneously or as the result of a mutagen is the addition of one or more nucleotides during DNA replication. Because translation of a gene begins with a specific codon and proceeds one codon at a time, the addition of an extra nucleotide shifts the codons in the mRNA. This type of mutation is termed a frame shift mutation. A frame shift affects all amino acids incorporated beyond the original site at which the addition occurred. If the new codon generated by the frame shift is a stop codon, the protein synthesized will be shortened and is often non-functional. Another type of frame shift mutation occurs when a nucleotide is deleted during DNA replication. Deletion of a nucleotide in DNA results in a change in the codons in messenger RNA from the point of the deletion and changes in the amino acids inserted into the protein.
Now, another type of mutation is called an inversion. And what happens in an inversion is that a piece of DNA is clipped out of the molecule and flipped front to back and then religated into the molecule. Now, you might wonder how in the world that could possibly happen. And one way that it can happen is through what are called mobile genetic elements. Okay, there are certain sequences of DNA that can encode for enzymes that clip that particular section of DNA out at specific base sequences and then insert it into a similar base sequence somewhere else in the molecule and then seal that insertion. Okay? Eukaryotes have these. They're called transposons. Okay? They are sometimes inverted repeats. They are sometimes not inverted repeats. It all depends on the type of element we're talking about. Sometimes the mobile genetic element encodes for the transposase and sometimes it doesn't. Okay? Sometimes inversions can happen independently of there being a mobile genetic element. A section of DNA can simply break off and then enzymes can attempt to reseal the injury using the broken segment and trying to patch it back in, or using the broken ends and then trying to basically copy over them and seal them. Okay? So there's all sorts of ways that this can take place. Base excision repair occurs when the damaged base is basically cut out and then a proper base is put back in. In methyl mismatch repair, methyl-directed repair enzymes search for the old strand and fix the damage in the unmethylated strand. Basically, the methylation is a way to chemically tag one of the strands of DNA as being a parent strand, okay, while the other strand is the newly reproduced strand, all right? And then there's SOS repair, coordinated responses that can introduce mutations to save the cell, and recombinational repair, which is replacement of damaged DNA through recombination mechanisms. Okay, so let's look at mutations and base excision repair.
Some damaged DNA bases are repaired by a system called base excision repair. In this system, specialized enzymes called DNA glycosylases recognize specific damaged bases and remove them from the DNA. The result is an intact phosphodiester backbone, missing a base. In the next step, enzymes called endonucleases cleave the phosphodiester backbone at the location of the missing base. The 3' end produced by the endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the missing base. DNA ligase then seals the remaining nick, and the repair process is complete.
So essentially what's going on here is that special enzymes recognize the damage in the DNA, removing it without breaking the phosphodiester backbone. The result is a site without bases in it that specific endonucleases can chop. After the excision and removal, DNA polymerase I makes a replacement strand on the proper base. Okay. We'll talk about plasmids and restriction enzymes and gel electrophoresis, and then we'll explain the formation of recombinant DNA molecules and talk about DNA hybridization and sequencing technology. Okay. Now, one of the reasons that we're so interested in bacteria in science is their use as um, basically a DNA factory, and also as a way to generate what are called constructs, which are pieces of DNA that are built from different sources. Okay, um, we can do this by modifying existing plasmids and putting in genes of interest that provide traits that we want to uh, include in the plasmid to make it easier to work with and when we study other pieces of DNA from other organisms. Okay, and we usually want to also have that plasmid be something that's screenable and selectable. Okay, where we can select for it using resistance to a drug such as ampicillin or chloramphenicol, or we can screen for it by the production of a colored reaction that occurs in the presence of a particular reagent, such as the use of the LAC-Z gene and a reagent called XGAL and an inducer called IPTG, where if the plasmid contains a piece of foreign DNA, the colony carrying the plasmid will grow up colorless on LB media, Luria broth media, or if it does not contain a piece of foreign DNA, it will be dark blue on that media. Again, using the XGAL and the IPTG in addition to the LB in the media. Okay, that's one way to determine whether or not a piece of foreign DNA has been cloned into your plasmid using tools called restriction endonucleases that cut DNA at specific base sequences, and DNA ligase, which seals those pieces of cut DNA from different sources together. Um, and the utility here is that using these restriction enzymes, which are actually sort of the immune system of the bacteria, okay, basically the way they work in the bacteria is that if foreign DNA from a bacteriophage or some other pathogen tries to come into a bacteria, it uses its restriction enzyme that the bacteria manufactures to chop that DNA up and ruin it so that it can't hurt the bacteria. Okay, and so a lot of these restriction endonucleases have names that indicate the bacteria that they come from. Okay, such as, for instance, HEY3 and Echo R1, right? HEY3 comes from Haemophilus bacteria. Okay, Echo R1 comes from E. coli. Okay, that's just a couple of examples. The way that a molecular biologist uses these tools is to cut DNA at recognition sequences because they know information about the DNA they're working with. They have a map of it that tells them where those restriction sites are, and that allows them to cut out pieces of DNA that are of interest to them and then seal them into a cut plasmid containing what are called compatible restriction sites, which would be sites with overhangs that can hybridize to each other, that can anneal, okay? And then we can seal those breaks with a ligase, for instance, where those arrows are located, and then make a hybrid molecule that comes from partly plasmid DNA and partly DNA from some other source, okay? The use then of that construct is that we can grow that plasmid with that piece of foreign DNA up in a bacterial culture, and then we can isolate large amounts of DNA from those transformed bacteria, and then we can put that DNA, that foreign DNA, to other uses. Okay, we can, for instance, add things to that foreign DNA so that we can see its expression in other types of cells, or we can modify that foreign DNA by introducing mutations or changes in it, and then we can introduce that foreign DNA into another organism and get it to integrate into the genome of that other organism and change the genetic composition, make a transgenic animal, for instance. Okay, those are just a few of the uses for these tools. So you could think of the restriction enzymes as special scissors that cut the DNA at specific sequences, and you can think of the DNA ligase as the glue or the paste or the tape that puts all of this back together. Okay? When you utilize restriction enzymes, one of the things that you can do with those restriction enzymes is visualize the fragments that result from cutting a DNA sample. Okay, the reason that you might want to do this, for instance, would be to check a plasmid that you've been given by another lab or from another source, or to check a piece of DNA that you've isolated from a different organism. Okay, and then what you do is you cut it with these enzymes, and you can determine from the fragments that are generated what the restriction map is. Okay, you can tell, for instance, if you're looking for plasmids that may or may not contain a foreign DNA fragment. Okay, that by cutting them with different restriction enzymes, what you predict the fragment sizes will be versus what the fragment sizes are in the actual electrophoresis. Now, the way the electrophoresis works is that you mix your cut DNA with a dye that lets it sink into a well. The gel itself is made of something called agarose, which is derived from seaweed, and it has the consistency of jello, okay, but it's not jello. Basically what it is a mesh of polysaccharide, and when you apply an electric field to it, the DNA molecules are attracted to the cathode because they have a negative charge, and the smaller molecules run faster than the larger molecules, and so you get separation based on size. Okay, and you can compare that to a reference DNA, a marker DNA that has fragments of a known size in it, and then you can determine the size of your different fragments and then generate a map of your piece of DNA. Okay, and that can tell you, for instance, whether or not you've got a foreign piece of DNA cloned into the plasmid or not. Okay, that would be just one use for it. Another use for it might be taking genomic DNA from another organism, like a plant or an animal, okay, and then cutting it with different enzymes, sometimes in combination, sometimes alone, and then running out a whole series of different fragments on a gel electrophoresis, and then you can actually probe for the fragments that contain your gene of interest using a piece of DNA from a known source that is complementary to your gene of interest, and you can tag that piece of DNA either radioactively or you can tag it with a dye. Okay, and then once you run your electrophoresis gel, you can stick those fragments in that gel to a nitrocellulose filter, which looks like a piece of paper. Okay, and you can attach it covalently to that filter using UV, and then you can turn around and probe with your special piece of DNA that's radioactive or that has a dye attached to it, and it'll only hybridize to regions of DNA on your nitrocellulose filter that contain areas of your gene of interest. Okay, and then that tells you a little bit about where your gene of interest is located, okay, and what the map of your gene of interest is. Okay, and you'd do that if you didn't have very much sequence information about your particular target. Okay, these days the databases for the DNA of a lot of different organisms is known, and so an application of gel electrophoresis might be to isolate a fragment containing your DNA from a restriction endonuclease digest. And because you know the predicted size, you can isolate the gel in the area of the size that you predict your gene of interest will be in, and then you can basically purify that DNA out of the gel and clone it into a plasmid, and then search for the plasmid from that cloning procedure that contains your gene of interest. Okay, so that's one approach. All right. You can see here an example of a restriction endonuclease digest, right? You've got your DNA that contains an echo site in it, another piece of DNA that does not, okay? And then you've got a mix of both, okay? And then you can determine what the map will look like with and without an EchoR1 site at that position. Now, what produces that EchoR1 site might be something like a mutation, okay? You could have a piece of DNA from a mutated and a non-mutated source, all right? And if that mutation produces a restriction site or destroys a restriction site, then you can determine using this type of analysis whether you have the mutant or you have what we call the wild type DNA. Okay? You can see here some examples of a restriction endonuclease digest. DNA samples are loaded into the different wells. You then apply the electric field, the negative electrode up here, the positive down here. You run the fragments through the gel with the smaller fragments going the fastest. And then you can stain the gel afterwards with dyes such as ethidium bromide or cyber green. And then you can pass a UV light through them, and the fragments will light up, and then you can take a photograph of it, which is what you're seeing here. Okay, and then you can look at the different sizes of the fragments and compare them to the marker and determine what the sizes of the fragments are. Okay, and then that can tell you information about your piece of DNA of interest. Generally, when you do an experiment like this, you always want to include one lane of DNA that is only cut one time, and also a lane of DNA from that sample that is not cut at all. Okay, and those serve as controls. All right, and then you can turn around and cut your DNA with different enzymes and get a restriction map. Okay, ribotyping of Haemophilus influenzae shows the relatedness of different strains of the bacteria based on what we call restriction fragment length polymorphisms. So the idea here is that you take the chromosomal DNA out of the bacteria, and then you cut it with a particular enzyme, and then you take that same DNA, that uncut DNA, cut it with a different enzyme, and then what you generate is a restriction map from different enzymes for different strains of the organism. Okay, and the reason we might want to do that is that one RFLP might correspond to a gene that produces a virulence factor in that bacteria and makes it a pathogen, right? Whereas if that RFLP isn't there, perhaps the virulence factor is also missing. Okay, so it's one way to sort of get an idea of what the DNA sequence looks like, sort of in a rough way. Okay, so you can use these sort of like markers. Okay, polymorphism just means different shapes or different appearances. Okay, this is a phylogenetic tree that was constructed by ribotyping 400 isolates of Haemophilus influenzae. The sequences were compared by computer analysis. The length of the horizontal bars represents the sequence difference between each pair of strains in base pairs per generation. The longer the bar, the more base differences separate the strains. Colored bars indicate letter types for strains that are encapsulated. Numbers at the right indicate new isolates of non-typable Haemophilus obtained in Finland from a group of children with otitis media, which is a middle ear infection. Okay? Otitis media is just what happens when bacteria get behind the eardrum and grow in the middle ear, which is the space between the inner ear and the eardrum itself. And as they do, they produce gas and pain and pressure, and that causes the eardrum to swell from the inside out, and it's extremely painful. Okay, and it can cause the eardrum to rupture, and if it does, you end up with scar tissue on the eardrum, and that can decrease your ability to hear well. Okay, and it's a problem with children because the pipe that connects the middle ear to the throat, which is called the eustachian tube, in children is very short and straight. And so when they get throat infections, or if they swim in contaminated water, then they can pick up those kinds of infections in the middle ear. Okay? Now, recombinant DNA is just like the name implies. You're recombining DNA from different sources to make what we call a construct. Okay, so what you do with a plasmid is you have a plasmid construct that you might buy from a company. Okay, and usually it'll contain an origin of replication that lets it reproduce itself in large numbers inside a bacterial cell. Usually the bacteria is E. coli. Okay, you'll have a selectable marker such as ampicillin resistance on that same plasmid. And then you'll have a screenable gene such as LAC-Z. In the middle of the LAC-Z gene will be a series of restriction endonuclease sites that allow you to clone a variety of different fragments of DNA from different sources. Okay, and those cut sites appear only once in the entire plasmid. Okay, so depending on the fragment that you're after, if you know something about your fragment, you can cut it with a single enzyme that cuts around your sequence and pop out a fragment that you can clone into the plasmid vector that's cut with the same restriction endonuclease. And because the single-stranded ends produced by the cut of the restriction endonuclease on both your plasmid and your foreign DNA are complementary, then they can anneal relatively easily. You just mix the fragments together, and then you can throw them into a ligation reaction. You just buy the ligase in a tube, and you add it to the fragment in the plasmid once you've heat and activated the restriction enzyme, and then it'll seal those pieces of DNA together, and then you can take that new construct and you can introduce it into new bacterial cells, E. coli cells, and they can generate large amounts of that construct for you, which you can then purify and then work with in downstream applications. Okay, so you can see here how the cleavage generates these sticky ends and how we can anneal them together. Sometimes, to make the cloning a little bit easier, you can cut the plasmid vector with two different restriction enzymes so that you can ensure that your fragment only goes into the plasmid vector in one orientation. Okay, and that's called unidirectional cloning. Sometimes, if the ends are the same, you can clone the fragment in either orientation. Okay, and that's bidirectional cloning. And so to determine what sort of insert you've got to cut your plasmid and then predict based on what your map says you'll get in either case, what your fragments will be on a restriction digest, and then you compare that to your prediction, right, and determine which of your colonies that grew up with your new construct inside it contains the plasmid that you're after, that has the foreign DNA in it that you want. Okay, the way that you transform bacterial cells, there's a variety of ways. You can use calcium chloride salts to treat the bacterial cells so that they're what's called competent, and then you can mix them with some DNA, your construct DNA, and then you can chill them on ice, and then you can heat shock them by moving them to 42 degrees centigrade for a short time, and then you can let them recover in ice, and then you can grow them in culture, and then once you've done that for about an hour at 37 degrees, you can plate those on selective media. That's one way to do it. The other way is through a process called electroporation, where you take bacterial cells that have been treated chemically a little bit differently, and you mix the DNA from your ligation reaction in there and then you have an electric current flow through a little chamber called a cuvette, and that causes little holes to open up in the bacterial cells, and they take up the DNA, and then you can grow them in culture for a bit so they can recover, and then you plate them on selective media. Okay, so those are just a few ways that you can go about doing this. Okay, sometimes you want to hybridize in order to detect a bacterial pathogen, right? You can take a probe that is complementary to pathogenic sequence, and then you can have your target DNA, and you can cut it and run it on a gel, okay? Or you can, in some cases, simply cut that DNA and UV link it to a nitrocellulose filter. That's another way to do it. And then you just apply your probe with your pathogenic sequence, and usually the probe is either radioactive or it has a dye attached to it. And then it'll anneal to target DNA if it's there. And then you can see which bands light up on a nitrocellulose transfer of your electrophoresis, or a nitrocellulose probe of just the DNA applied to little round wells. Basically, you can take the nitrocellulose, you can cut it about the size of a playing card. You can put it over some filter paper that has a vacuum on it. You can take then something that looks like basically a plastic plate that has a lot of little holes in it that are arranged in a specific order, okay? And then you basically suck that DNA onto that nitrocellulose filter, but it won't go anywhere but in the little round wells where you put it. And then you can let that filter dry. You can treat it with UV to covalently link the DNA there, and then you can probe the DNA with your probe, okay? And only those wells that light up will be the wells that have your DNA targeted. That's called a dot blot, okay? These are all variations of a process known as Southern blotting, which was developed by a guy named Southern, where you use DNA transferred onto a nitrocellulose filter, and then you probe it with something. It could be RNA, it could be DNA, as long as it's a nucleic acid, okay? And the basis for the process is that the complementarity of the probe and the target means that the probe will stick where the target is and won't stick anywhere else. Okay? The PCR is another technique that we use, and it's actually the most popular technique today for doing molecular biology, because you can make large amounts of DNA of a desired sequence from a very small sample, okay? And so you don't really have to worry about things like cutting DNA up or probing DNA to find your sequence of interest. Instead, you can use your primers, which are complementary to regions that are in front of and behind your gene of interest, in order to produce large amounts of DNA from your source. Okay, you can check whether or not the reaction worked using gel electrophoresis, or in some cases you can do something called real-time PCR, where you could actually see the reaction in real time by having the DNA product, as it builds up in the tube, grab a dye that's also in the tube, and then the reaction chamber will glow, and then you can see that on a readout on the machine, okay? Or you can do the old-fashioned thing and just run it on a gel and look at it, okay? And then you can take that PCR product and you can clean it up by removing extra primer and any unincorporated nucleotides. And then, usually because people design primers to have restriction sites of their choosing at either end, you have a custom piece of DNA that you can clone into a plasmid that might become part of a larger construct. Okay, but you can also use PCR to just detect sequences of interest in a sample. Okay, and more than one sequence of interest in many cases. You can use whole sets of primers, like 3, 4, 5, 6, 7 sets of primers at the same time, and then generate sequences and see whether or not you've got your sequence of interest. And in some cases, you can even have those primer sets have different dyes on them, so that you end up producing different products that glow a different color when you hit them with a laser beam or with UV light in some cases. Okay, and you can tell which sequences you've got in that sample. Okay, so this is PCR.
The polymerase chain reaction, or PCR, is a technique that can produce over a million-fold amplification of target DNA within a few hours. PCR requires specific oligonucleotide primers and plenty of nucleotides for DNA polymerization. The DNA polymerase must be heat stable because PCR requires repeated cycles of heating the solution to 95 degrees Celsius. Heat-stable polymerase is obtained from thermophilic bacteria or archaea. In PCR, the reaction mixture is heated to 95 degrees Celsius for 30 seconds to denature or separate the DNA strands. The temperature is then lowered to 55 degrees Celsius for 30 seconds, which allows the oligonucleotide primers to anneal to known DNA sequences flanking the target DNA. The oligonucleotide primers, which are usually 20 to 30 nucleotides in length, are made so that they are complementary to the flanking sequences. The temperature is then brought to 72 degrees Celsius, which is the optimal reaction temperature for the heat-stable DNA polymerase. The polymerase uses the primers as starting points for DNA synthesis, adding one nucleotide at a time to create a complementary strand of DNA. The cycle is repeated, starting with the 95 degrees Celsius heating step. Cooling the solution to 55 degrees Celsius allows the primers to anneal. Heating the solution back to 72 degrees Celsius allows polymerization to proceed. After 2 cycles, 4 double-stranded target DNA sequences exist. After the 3rd cycle, 8 copies exist. After the 4th cycle, 16 copies exist. After 25 cycles, about 30 million copies of the target DNA exist. Because PCR reactions require 25 to 30 heating and cooling cycles, a machine called a thermocycler is used to reproducibly and rapidly deliver these cycles.
Now, the reason that PCR is possible is because we've discovered and purified an enzyme called Taq polymerase from an organism called Thermus aquaticus that lives in hydrothermal vents down in the ocean floor at temperatures that are about that of boiling water and a pH close to that of battery acid. And Taq, Thermus aquaticus, loves those conditions, and the tools of life, the proteins that that organism uses to live in that extreme environment, also like those conditions. And so when we do PCR, we do cycles of heating and cooling inside something called a thermocycler. And the reason that we don't have to worry about adding new enzyme is because the Taq polymerase can handle the changes in temperature without having the enzyme be ruined. Okay? That enzyme, in fact, was discovered and characterized at the University of Cincinnati by someone I knew during my graduate work by the name of John Trella. Okay, the process of PCR was developed by a company called Perkin-Elmer Cetus. Okay, which no longer is involved in PCR. It's gone on to other things, but they patented the process. Now, DNA sequencing is a way to determine what the arrangement of bases is in the DNA double helix. Okay, so this kind of is the primary structure of the DNA. And what you do here is we utilize two different methods now of DNA sequencing. One is called Sanger dideoxy sequencing, and then the other um, is what's called deep sequencing. Okay, so the Sanger dideoxy sequencing, which is something that I've done in the past and that I've used in the past, involves the use of what are called dye terminators. So what you do there is you take your DNA sample, all right, and you provide or you get a primer that sits upstream of the area that you want the sequence of. The primer will sit down in front of the sequence you're interested in, and then you throw in all the nucleotides needed to build DNA, along with special dye terminators, which are dideoxynucleotides that have specific dyes attached to them that glow a specific color when they're hit with a laser beam. Okay, so you get dideoxy A, G, C, and T. They're all different fluorescent dyes attached to each of those, and then you throw those into the reaction along with the regular nucleotides, and then what will happen is that you'll generate a random array of products that all terminate with a dideoxy, all right, that's complementary to the template strand. And from that, you can develop the sequence of the complementary strand, okay? And that tells you the sequence of the region that you're interested in, okay? If you end up getting sequence information that doesn't go far enough into your area of interest, you can always design another primer that sits within the area of known DNA sequence and then continue the sequencing process until you get to the other end. And you can also sequence from both ends of your gene of interest if you like. Okay? And then eventually what you do is you get a read that goes all the way across your gene of interest, and then you have that sequence information. Okay? Now, why would you want to do this? Well, you might want to compare that sequence information to a database to see what it is that's in your piece of foreign DNA, right? Are there genes in there that have already been characterized, or do you have a unique arrangement of genes that hasn't been seen before, or are your genes similar to other known genes, okay? The other reason that you use DNA sequencing is to check constructs before you use them for downstream purposes to make sure they don't have any goofs in them, okay? So you might have built something by cloning DNA together. Maybe you generated a construct that you wanted to use to make a transgenic mouse, all right? And you've got your DNA that you put in there, and you checked it by restriction mapping, gel electrophoresis, maybe even did some PCR on it to check. And all that looked pretty good, but to get it perfect, you want to know really from base pair to base pair what you have. And so you would get a set of primers that would allow you to walk across the entire region of the construct that you built to make sure that the sequence matched what you thought it matched, that there weren't any mistakes in there that happened during the cloning process or during the time that you grew it in the bacteria. And only then would you use that construct to make a transgenic animal, which can be an extremely expensive procedure. Okay, the reason we make transgenic animals, one reason is to recreate conditions that mimic human diseases, for instance. Okay, so we might see a human disease that corresponds with a mutation in a certain human gene. All right, and then we might look in the mouse for the similar gene. Okay, and then we might generate a deliberate mutation in that gene that's like the mutation in the human gene, and then we would generate a mouse from that that had that mutation, and then we could see whether that mutation produced similar changes in the mouse. Okay, that's just one application that you could use it for. Okay, the other way you might use it is to sequence areas of DNA that come from cancer tissue, so that you can detect areas of mutation that might have led to the change from a normal to a cancer cell in that particular tissue. Okay, so there's all sorts of possible applications for DNA sequencing. And it's gotten so inexpensive now that we can sequence the genomes of whole organisms very inexpensively. Okay. The way this works for dideoxy is shown here.
Much of the progress that has been made in molecular biology in recent years has been in large part due to the ability to sequence an organism's DNA. Although large-scale efforts of this sort, such as the Human Genome Project, have used more advanced technology, basic DNA sequencing capability arose out of the dideoxy sequencing method. This method is therefore foundational and is the subject of this video. To understand the dideoxy sequencing method, it is important to have a firm grasp of basic DNA structure. DNA stands for deoxyribonucleic acid. It is assembled from individual nucleotides. A nucleotide has three distinct parts: the phosphate group, the sugar, in this case deoxyribose, and the nitrogenous base, which could be A, G, C, or T. In this case it's T, thymine. To link individual nucleotides, a bond must form between the phosphate group of one and the sugar group of another. More specifically, the hydroxyl groups from the phosphate and the sugar combine to form a molecule of water, which is produced as the bond is formed. This process is known as dehydration synthesis because water is the byproduct. Now remember that with DNA, the sugar is deoxyribose. This name comes from the fact that one hydroxyl group has already lost its oxygen. If we replaced deoxyribose with dideoxyribose, both hydroxyl groups would be missing their oxygen. In this case, an adjacent nucleotide would be unable to bond because dehydration synthesis cannot occur. This sequence stopping power of a dideoxy nucleotide is at the core of dideoxy sequencing. Building on this stopping power, we must remember that there are four distinct nitrogenous bases: thymine, cytosine, adenine, and guanine. Therefore, as we attempt to build a DNA molecule, it is possible for us to employ dideoxy nucleotides for only one type of nucleotide, either A, G, C, or T. Therefore, we could stop DNA sequence construction only at sites where that particular base appears. We can use this stopping power to decode a DNA sequence. In our first experiment, experiment T, we'll use regular deoxynucleotides for A, G, and C. However, for half of the T nucleotides, we'll use a dideoxy form, giving the possibility of stopping the DNA sequence assembly. If we do this experiment enough times, we'll obtain each of the following varieties of sequences. It might be the case that even though half of the nucleotides are dideoxy, none of them ever get inserted and we get the full sequence anyway. Or, perhaps, we insert a dideoxy T here and cut the sequence short by one. Or maybe we insert the dideoxy T1 earlier and cut the sequence still shorter. Finally, it's possible that the first nucleotide we add is dideoxy and the sequence stops at 1. Therefore, each of these four possibilities could exist when we use dideoxy T. Now, let's do another experiment. In this one, experiment C, we'll use regular deoxynucleotides for A, G, and T, but half of our C nucleotides will be dideoxy. We do the experiment a number of times and observe the following forms. Again, no dideoxys included. The full sequence is obtained. We might, however, insert a dideoxy C in the first C position. Because C is at the final position of the sequence, it's difficult to determine if there is in fact a C there or if we've just gotten the full-length result. We can't exactly know for sure. Let's do the same experiment for G. In this case, again, it's possible that we get the full sequence. Or it's possible that at our first instance of G, we insert a dideoxy and the sequence stops. Therefore, there are two options for our G experiment. Finally, we'll repeat the experiment for A, considering the possibility that we get the full sequence, or that at our first instance of A, a dideoxy nucleotide is inserted and the sequence stops. However, you may be wondering how we actually analyze DNA sequence length. We can't exactly use a ruler. Instead, we will make the DNA fragments have a race through a thick gel. Because we can't count on DNA to run by itself, we'll use an electrical current to pull the DNA. This process is called gel electrophoresis. Small DNA molecules will travel furthest through the gel, while large molecules will have a hard time making progress and will therefore stay closer to the starting point. We will put the DNA molecules in lanes according to which dideoxynucleotide we were using for a given experiment. For experiment T, we'll put all of those results in a lane labeled T. Now we'll let the gel run. Using the knowledge that the smallest sequences would probably make the furthest progress, and by considering the results of our race, we can reconstruct the sequence of nucleotides used to build this DNA sequence. The group traveling furthest was the smallest, 1 nucleotide. Because it's in lane T, we can check back and put a T in position 1. We can use this same process of analysis to reconstruct the entire sequence. Our next furthest DNA sequence comes in lane G, so we put a G into our sequence. A, T, and T. This gives us a sequence of TGATT. What about our last position, though? The position occupied in all the lanes? Well, because it's possible to build the full sequence without ever inserting a dideoxy stopper nucleotide, this sequence will appear in each lane. Therefore, we can't determine which nucleotide is in the final position. However, if we really wanted to know this base, all we would have to do is examine a bit longer segment. Now you have a basic understanding of the technique that was fundamental in the modernization of genetics, molecular biology, and the study of organisms' genomes.
Okay, we'll talk about different kinds of RNA and how they're made from DNA. We'll talk about how RNA polymerase does transcription, and then we'll talk about reverse transcriptase and what a cDNA is. Okay, so the central dogma of biology simply describes the flow of information in a typical cell, which is from DNA and then through transcription to messenger RNA, and then via translation we use that information to produce the protein, and then the protein goes out and does the task. Okay, and so in a prokaryote, this happens all in the cytoplasm because transcription and translation happen in the same physical space. Okay. In a eukaryote, the transcription happens in the nucleus, and the translation happens either on the rough endoplasmic reticulum or in the cytoplasm. Okay? You can see here an overview of gene expression, where you've aligned here a bacterial structural gene with its message. RNA polymerase generally binds the -10 and -35 regions of the DNA, and then reading the template strand begins RNA transcription at the area indicated as +1. Transcription makes the messenger RNA, which has an untranslated region of variable length and then a ribosomal binding site. A few nucleotides downstream, the ribosome begins translating at the start codon, which is usually an AUG. The start codon specifies the amino terminus of the translated protein, which is always made by the ribosomes in the amino to carboxyl direction. Okay? So basically, what are we saying here? All right? We have a gatekeeping region, the promoter. Okay? What will happen is RNA polymerase will bind here and then will begin making message from +1 all the way down to the transcription terminator. All right? Once it gets there, what will happen is that it'll let go of the message, okay? And then ribosomes will bind the 5' end and then scan for a starter AUG, which they will then use to walk down the structural gene, okay, until they get to a translation stop. They'll release the protein, and then an internal ribosome binding site called an iris will let other ribosomes come in on the same transcript and do the same thing for the next gene, okay? And then it'll let go at another stop codon. So what you get here is some economy of gene expression, right? In the same transcript, you get to translate two proteins that are generally related in function to some biochemical pathway that takes place inside the cell, okay? So let's take a look at transcription.
The first step in gene expression is to make an RNA copy of a gene's DNA sequence. That is, to transcribe a gene. Transcription includes the stages of initiation, elongation, and termination, and is performed by an enzyme called RNA polymerase. In bacteria, before initiation, RNA polymerase includes a number of subunits, including one called sigma. Sigma factor binds tightly to promoters, which target the polymerase to a gene. With the sigma factor attached, the composite RNA polymerase is known as a holoenzyme, meaning that it includes all the subunits needed for initiation. The holoenzyme form of RNA polymerase binds loosely to any sequence of DNA. Thus, within the cell, the RNA polymerase binds and comes off DNA rapidly, as if scanning the sequence. When the RNA polymerase binds to a promoter sequence, however, the binding is tight. To start transcribing a gene, the RNA polymerase opens the DNA, creating a transcription bubble. Now the RNA polymerase can fit a ribonucleotide to base pair with the DNA. So begins the elongation stage of transcription. To elongate the chain, the polymerase catalyzes the addition of new ribonucleotide triphosphates, RNTPs. Each incoming RNTP condenses with the 3' hydroxyl of the preceding nucleotide, releasing pyrophosphate in a step that yields energy. Only a few ribonucleotides can be added before the polymerase stops, because the whole complex is still tightly bound to the promoter. However, now the sigma factor falls off the complex, allowing the core RNA polymerase to continue to move along the template, synthesizing RNA at approximately 45 bases per second. Finally, transcription must finish at the end of the gene, at a pause site in the DNA sequence that causes the RNA polymerase to fall off. In bacteria, even while transcription is occurring, the next stage in gene expression, the translation of a protein, also begins. Here, RNA polymerase is transcribing a gene. The beginning of the growing mRNA has a region called a ribosome binding site, where a ribosome can bind and start translation. As the mRNA lengthens, additional ribosomes can hop on, creating a polysome, which is an mRNA with a series of ribosomes translating the sequence into proteins. This electron micrograph shows a polysome. Notice how the ribosomes farthest from the 5' end, the beginning of the mRNA, show the longest growing peptide chains.
Now, occasionally what will happen is that we'll be interested in producing a DNA copy of a messenger RNA to use for other purposes. The reason we might want to do this is that we might want just the coding regions of the gene to work with, okay? RNA is notoriously chemically unstable and easily degrades in solution. And so by taking the information in the messenger RNA and transferring it to DNA, we get a more stable molecule. Reverse transcriptase is an enzyme that's carried by a type of bacteriophage called, or a type of virus called a retrovirus. Okay, the way retroviruses work is if they come in and invade a cell, they have RNA as their genetic material. The reverse transcriptase turns around and makes a DNA copy of that RNA, and then that DNA copy goes to the nucleus and is used to make many, many copies of RNA that ends up as the genome for new virus particles. Okay, so that's the utility to the virus. What we use it for, what molecular biologists use it for, is to make stable copies of messenger RNA that can then be cloned into a construct and then modified for downstream applications. Okay, and so that's called a cDNA. Right, a cDNA is going to be just the coding regions of the gene and nothing else. Now, in a eukaryote, the cDNA thus won't contain the untranslated regions or the introns. Okay, all it will contain is everything from +1 through all the coding regions to the polyadenylation site. All right, and in the early days of transgenic animals, we used to very simple-mindedly make cDNA copies of genes as a way to study how genes work. And we would take these constructs and put them into animals and make transgenic animals, and sometimes the constructs would work the way we predicted and sometimes they wouldn't. And it turns out that frequently the reason that the constructs didn't work the way we predicted is because a lot of gene regulation in eukaryotes is actually contained in these intron regions, which are removed in a messenger RNA in a eukaryote. Okay, so that caused us to kind of go down a blind alley in many cases. It wasn't until the discovery of regulatory sequences in introns that we sort of changed our tune after that. Okay. What we're going to do next is talk about transcription and translation in bacteria, talk about the ribosomal steps of translation, okay, and how we can target them with antimicrobials, antibiotics, and then we'll explain how proteins end up needing to find their native conformation in order to function. Okay, so let's take a look at translation.
Protein synthesis occurs in 3 phases: initiation, elongation, and termination. In bacteria, such as E. coli, initiation requires the association of several components: small proteins called initiation factors, the first tRNA, the mRNA to be translated, and the small subunit of the ribosome, also called the 30S subunit. The key event of initiation is the association of the mRNA and 30S subunit so that the start codon, AUG, is centered on the ribosomal subunit. The 50S subunit docks to complete the initiation complex. Initiation uses energy by hydrolysis of GTP. Note that the ribosome has 3 tRNA binding sites: an acceptor, A site, also called aminoacyl site, a peptide, P site, and an exit, E site. The ribosome is ready for elongating a chain of amino acids. Elongation requires an elongation factor called EFTU, associated with GTP, to bind to aminoacyl tRNAs. This complex enters the A site. Correct selection of the tRNA complex depends on codon-anticodon pairing. Here the anticodon CCC pairs with a GGG codon. GGG codes for the amino acid glycine, which this tRNA carries. Next, the ribosome catalyzes the formation of a peptide bond between the 2 amino acids. and the hydrolysis of GTP drives peptide bond formation. The resulting EFTU, GDP, is expelled. An elongation factor called EFG, associated with GTP, binds to the ribosome. The GTP is hydrolyzed, providing the energy to ratchet the 50S and 30S subunits ahead one codon. This maneuver opens up the A site, and the uncharged tRNA slides into the E site. The next aminoacyl tRNA that enters the A site creates a conformational change in the ribosome that telegraphs through to the E site and ejects the uncharged tRNA. These elongation steps repeat along the mRNA. Eventually, the ribosome arrives at the end of the coding region, marked by one of 3 possible stop codons. This phase of translation is called termination. No corresponding tRNAs exist for stop codons. Instead, a protein called a release factor, which has the general shape of a tRNA, enters the A site. The release factor triggers hydrolysis of the peptide tRNA bond, thus releasing the peptide. The 30S and 50S subunits fall off the mRNA and separate in the cytoplasm, available once again to form a new translation complex with another mRNA.
So you can see here some of the different types of antimicrobials that are available and what they target. Okay, the reason this works for treating disease in humans is because we generally target aspects of prokaryotic reproduction that don't have a counterpart in a eukaryotic cell. Okay, an example would be the prokaryotic ribosome. Right, the prokaryotic ribosome is functionally identical but structurally different from the eukaryotic ribosome. So a drug that targets bacterial ribosomes is going to hurt bacterial reproduction way before it hurts the health of the cells in our body. Okay. Eventually, though, you can reach a level where the function of the cells in our body will be affected because it could end up hurting mitochondrial function. Okay, but usually it takes a much higher concentration to do that to a mitochondria than it does to do that to a free-living bacterial strain. And you can see that a lot of these antimicrobials attack the ribosome. Okay, they bind it and they reduce its ability to do the translation job properly. All right. Unfortunately, the minute you start using antimicrobials, you're applying selection pressure to the microbes that are out there, and you'll eventually select for microbes that are resistant to those antimicrobials, and oftentimes those resistance genes lie on plasmids that can be transferred from a resistant strain to a non-resistant strain. And if this happens many, many times, you can end up with what are called superbugs that are resistant to many different antimicrobials at the same time. Okay? We'll talk about how and why a cell regulates the expression of virulence genes, and we'll outline what gene regulation is all about. Okay? Some proteins, like RNA polymerase, are always needed for growth. Without it, the cell cannot reproduce. Most proteins are needed only under a limited set of conditions, and so to compete successfully with other organisms, microbes can't waste energy making proteins when they don't need them. Okay? So not only do we have to have economy of information in the bacterial DNA, we need to use these tools when they're absolutely necessary, and then we need to turn them off and turn off production of those tools when they're not needed, or else the bacteria is going to waste time doing something that's totally unnecessary. Okay? So how is it that we can regulate gene expression? Well, we can change DNA sequence, right? Some microbes can change DNA sequence to activate or disable a particular gene. One aspect of that is something called phase variation. All right, where literally we change the primary sequence in the double helix. We can control transcription by regulating it with repressors and activators and different sigma factors. We'll learn about that in just a second. We can do translational control, or we can control transcription, can introduce secondary structure in the message that can have an effect on the ability of the ribosome to translate, okay? And then we can also have transcription initiation sequences that recognize repressor proteins, okay, that can inhibit the ability of the gene to be translated. And then finally, in post-translational control, we have control of proteins already made that can activate, deactivate, or degrade the protein after it's been whipped out. Okay. Example here, right? The regulatory gene encoding a regulatory protein can be located near or far away from the target on the chromosome. When it's located nearby, it's usually transcribed separately from the target. The regulatory protein will stick to DNA sequences near the promoter of the target and control whether or not transcription is going to happen. Okay, so it'll either block or not block the ability of the RNA polymerase to go from the promoter through the structural region and then eventually to the terminator. Okay. Repressor proteins bind to DNA sequences and prevent transcription. One type of repressor binds to DNA to prevent transcription. To induce transcription requires repressor binding an inducer molecule. A different type of repressor has to bind to a chemical ligand called a co-repressor before the repressor can attach to DNA. Without the co-repressor, the gene will be transcribed. Okay, so there's different ways that we can turn the switch on and off. Okay, one that has the repressor usually on and the gene usually turned off is called an inducible operon. Okay, whereas one that requires a co-repressor is called a repressible operon. Okay, so one of these inducible operons is called the lac operon. It's well studied, well characterized. So let's take a look at the lac operon.
Many bacteria grow on glucose before they utilize other compounds, such as lactose, as a growth substrate when both are present in the medium. Transcription of the lactose operon is controlled by two regulatory proteins. The catabolite activator protein can bind to the activator binding site and facilitate transcription, and the lac repressor protein can bind to the operator site and block transcription. The catabolite activator protein can bind to the activator binding site only if cyclic AMP is bound to it. When glucose is present, the concentration of cyclic AMP in the cell is low. RNA polymerase cannot bind efficiently to the promoter, and transcription does not occur unless the catabolite binding protein is bound to the activator binding site. Therefore, transcription of the lac operon does not occur in the presence of glucose. In addition, if there is no lactose in the medium, the lac repressor binds to the operator site, and this blocks transcription. When both glucose and lactose are present, there is insufficient cyclic AMP to bind to the catabolite activator protein, which therefore cannot bind to the activator binding site. Without the bound activator protein, RNA polymerase cannot bind to the promoter and initiate transcription, even though lactose binds to the repressor and prevents it from binding to the operator site. When neither glucose nor lactose is present, the concentration of cyclic AMP is high, and the catabolite activator protein is bound to the activator binding site. RNA polymerase can bind to the promoter, but it is blocked by the repressor on the operator site. When glucose is absent and lactose is present in the medium, cyclic AMP is present and binds to the catabolite activator protein, which in turn binds to the activator binding site and facilitates transcription. Lactose binds to the repressor and prevents it from binding to the operator site. RNA polymerase can now bind to the promoter and carry out transcription.
The expression of genes can be transcriptionally regulated. Such regulation is a critical feature of both eukaryotic and prokaryotic organisms. One of the classical systems used to investigate transcriptional regulation was originally investigated by Jacob, Monod, and Pardee. Their studies involved the transcriptional regulation of the genes used to ferment lactose in the bacterium Escherichia coli. 3 genes clustered together on the chromosome are required for the bacterium to utilize lactose. These were determined to be the LAC-Z gene that codes for the enzyme beta-galactosidase, the LAC-Y gene that codes for a lactose permease, and the LAC-A gene that codes for the enzyme galactoside transacetylase. The LAC-P region is the promoter that is needed to transcribe the LAC-Z, LAC-Y, and LAC-A genes as a single polycistronic mRNA. The LAC-O region is an operator site that is involved in the transcriptional regulation of the LAC operon. The unit consisting of the LAC promoter, LAC operator, and the LAC-Z, LAC-Y, and LAC-A genes is called the LAC operon. Located near but not in tandem with the LAC region is the LAC-I regulatory gene that codes for an mRNA that is translated to produce a protein referred to as the LAC repressor. In the absence of lactose inside the cell, the LAC repressor protein is active. In its active state, the LAC repressor recognizes and binds to the LAC operator site. When the LAC repressor is bound to the LAC operator site, RNA polymerase is prohibited from recognizing the LAC promoter, and the LAC-Z, LAC-Y, and LAC-A genes are not transcribed. When lactose enters the cell, a small amount of it is converted to allolactose via beta-galactosidase. Allolactose binds to the LAC repressor. This causes a conformational change in the LAC repressor protein that prevents it from binding to the LAC operator site. Inactive LAC repressor is unable to bind to the LAC operator site. Without an active LAC repressor bound to the LAC operator site, RNA polymerase is able to bind to the promoter, and a polycistronic messenger RNA is transcribed that includes the LAC-Z, LAC-Y, and LAC-A genes. This polycistronic messenger RNA is then translated to produce the beta-galactosidase, lactose permease, and galactoside transacetylase proteins. Therefore, the induction of the LAC operon enables the bacterium to efficiently transport lactose into the cell and metabolize it.
Induction of transcription can involve an activator protein, which has to bind to a specific chemical ligand. This happens in the cytoplasm before the protein can bind to a DNA sequence near the target gene. The binding activates transcription on the target gene. Okay? So basically what happens is that once the inducer hits the activator makes the promoter more available to the RNA polymerase. Okay? So it's sort of like opening the door for the RNA polymerase to walk down the gene. Okay? An example, right? Corynebacterium diphtheria will usually encounter sufficient iron for growth, so it doesn't need to produce diphtheria toxin. But in human hosts, the iron is inside the host cells in the erythrocytes bound to something called hemoglobin. So diphtheria toxin will destroy those cells and release iron that the bacteria needs, and that's called hemolysis. All right? So what happens when the iron levels are low, okay, is that the RNA polymerase is going to end up binding to the promoter in front of the diphtheria toxin gene. The diphtheria toxin gene will get made, the message will get made, will make the diphtheria toxin, and then will lyse those erythrocytes and free that iron, and then those cells can grow. And the reason for this is that at low iron levels, right, the the repressor is not bound. Okay, so this isn't blocking access to the promoter anymore. Okay, at high iron levels, what happens is that the repressor and the iron together binds to the diphtheria toxin that is on the regulatory region that's blocking the ability of the polymerase to move from the promoter through the structural portion of the gene. Okay? Another way that we can do this, all right, is through environmental signals that use cell signaling, okay, or second messenger. A transmembrane sensor kinase protein picks up an environmental condition outside the cell. Binding the ligand triggers phosphorylation of a sensor kinase. The phosphorylated kinase then transfers a phosphate to a response regulator protein in the cytoplasm. The response regulator then binds the target operator or activator DNA and inhibits or stimulates gene expression depending on the gene. To downregulate the system, an enzyme removes the phosphate from the regulator. Okay, so this is what's called a cascade. So we bind this ligand, we then end up activating a membrane protein, which then phosphorylates a protein, which then phosphorylates another protein, which then eventually binds the regulator region and either turns on or turns off the gene. Okay, now you might wonder why so many steps in this process, and the answer is that at each step it's not a one-to-one correspondence, right? We can basically flip on this membrane protein and it can activate several of these response regulators, which in turn can activate several of these activators or inhibitors, and the result then is that your target is going to respond either by turning on or turning off very quickly. Okay, so it amplifies at each step. I like to think of it a little bit like reverse freeze tag. I don't know if you ever played freeze tag when you were little, but in freeze tag, one person was it. Everybody they touched froze, and when you froze everybody, you won, right? Well, think of that like in reverse. You're it, everybody you touch is it, everybody they touch is it, and pretty soon everybody is it, right? And the result is that the endpoint gets reached really fast, okay? And then once we do our job, once we fill the quota for that gene, for that particular response, then we're going to end up adapting to this new signal in some sort of way. And at that point, there will be enzymes that will remove these phosphate groups, and then these proteins will calm back down. Okay, and so that's called feedback inhibition. All right, and cascading, right? Signal cascades and inhibition are important ways to control cellular homeostasis, right? Which is the ability of the cell to maintain conditions compatible with life, even though the environmental conditions are changing all the time. Okay, so these are the videos that were incorporated, a few of them, right? So make sure if you haven't watched them in the course of this lecture that you go back and review them, okay? And with that, I will see everybody in the next podcast. I thank you for listening.